Hello everyone! Today we are going to talk about nitrogen metabolism as covered in Chapter 18 of our textbook. So today we want to hit the highlights of both anabolism and catabolism. So anabolism, we can talk about nitrogen fixation and assimilation, amino acid synthesis, and protein and nucleic acid synthesis. So we're just going to hit the highlights today because our time is short. So if we look at our overview of anabolism, we can see that we are here where we can use nitrogen compounds to make amino acids. Of course, we should note as mammals that we cannot fix nitrogen. We need bacteria to do that. And then we can use the amino acids to synthesize proteins. We should also keep in mind that amino acids and ammonium ions are used to make nucleotides to serve as the raw materials for DNA and RNA synthesis. So let's first start with nitrogen fixation. So nitrogen fixing bacteria have a nitrogenase enzyme, and that allows them to convert nitrogen to ammonia. And that is reducing the nitrogen, so we need a lot of protons and electrons and quite a lot of ATP because this is a very uphill reaction. So this allows the nitrogen, remember from general chemistry that nitrogen has a triple bond, so that allows the triple bond to be broken and protons and electrons to be donated to the nitrogen to form ammonia. We have nitrates present in water and soil. This happens when lightning, which provides quite a lot of energy, can provide sufficient activation energy for nitrogen and oxygen in the air to be converted to nitrates. And then they rain down into the water and the soil. And then the nitrates can be reduced to ammonia by plants, fungi, and bacteria into enzymatic steps. So this is summarized here where we look at our nitrogen oxidation state on this scale right here, where we go from most oxidized to the nitrates to the ammonia and ammonia ions, which are the most reduced. So the red arrows show that those reactions largely occur in anaerobic environments, but you notice that all of these reactions occur in bacteria, fungi, and archaea. So we as humans are quite reliant on these organisms to help us out, because the only way that we can get nitrogen in order to build the protein components of our bodies is to ingest amino acids through either eating plants or eating animals. So we are not able to fix the nitrogen ourselves. So from a dietary standpoint, we can take the 20 amino acids and we can classify them as essential or non-essential amino acids. So essential amino acids are those amino acids that we cannot synthesize on our own and must obtain in our diet. So the thinking is that these pathways that microorganisms and plants have were lost in higher organisms during evolution. After all, it makes more sense from an energy point of view that you can just eat a plant and get your amino acid that way than devoting the metabolic energy that you need to make it yourself. So once we have a small amount of these essential amino acids, then we can take those and we can convert those to non-essential amino acids from those amino acids and glycolytic and citric acid cycle intermediates via transamination reactions. So we want to talk about that next. So transamination reactions are used to synthesize non-essential amino acids. So we can take an amino acid and an alpha keto acid, and we can transform them into a second amino acid and a second alpha keto acid. So the keto acid becomes a new amino acid, and the amino acid forms a new alpha keto acid. The enzyme is a transaminase or amino transferase, they're the same thing, and the vitamin required is pyridoxal phosphate, which is vitamin B6. So how do these reactions take place? Let me get my pen tool here. So if I look at the amino acid, so we know we have 20 amino acids and we have learned all of their R groups, but probably haven't thought about this term alpha keto acid. So over here in yellow, this is the acidic part, that's a carboxylic acid. And in the older nomenclature, the next carbon in is the alpha carbon. So the alpha carbon has a ketone group, so this is an alpha keto acid. And then that means that this is the rest of the molecule, so we will call this the R group too. And in a transamination reaction, what you do is you just trade R groups. So the amino acid over here gives its R group to the alpha keto acid, and then the amino acid takes the R group from the alpha keto acid. So we now have a new amino acid and a new alpha keto acid. So let's talk about how we can use these to synthesize amino acids. So if we have pyruvate, again, maybe we didn't think about pyruvate as an alpha keto acid, but it is. Here's the carboxylic acid group right here, and then here's the alpha carbon right here, and the R group for pyruvate is a methyl group. So that means that when we do a transamination reaction, some other amino acid will carry out this the trading of the R groups, and then the new amino acid we get is alanine. So in one step, we can go from pyruvate to alanine using a single transamination reaction, and we can also use pyruvate as a starting material for valine, leucine, and isoleucine, which are the branched chain amino acids. So we can use this as an opportunity to digress a little bit and talk about how we can use this transamination reaction as part of a larger cycle called the glucose-alanine cycle. So here, glucose is converted to pyruvate. Pyruvate is then converted to lactate in the muscle when oxygen is in short supply. The involvement of a transamination reaction right here, and this can go in both directions, can produce pyruvate from alanine. Or we could go the other way. Pyruvate can be converted to alanine. The alanine then can leave the muscle and go into the liver. Deaminating the alanine produces ammonia, which can be converted to urea in the urea cycle and then excreted. But what we're left here is pyruvate, and pyruvate can be converted to glucose via gluconeogenesis, which the liver can do and the muscle can't. And then the glucose then is shipped off to the muscle to generate more energy. So what you should notice about the cycle is that glucose is generated at the expense of body protein. So let's look at some other transamination reactions involving other compounds that we might not have thought about as alpha keto acids. So OAA is one of them. So here's the acid functional group, and here's our alpha keto group right here. So that means that this is the R group. So the R group, the CH2COO- is of course the R group in aspartate. So we can carry out a transamination reaction using an aminotransferase enzyme where we take the R group of oxaloacetate and we can convert it to aspartate, because again this would be the R group of the alpha keto acid, and then this is the R group of the aspartate or the amino acid. And then from there, so we have OAA is converted to aspartate in the single transamination reaction. From there, it is possible to convert aspartate into asparagine, methionine, lysine, and threonine. Here's another reaction. This is alpha ketoglutarate, and that might be a little bit more obvious. That's an alpha keto acid. So again, here's the acid functional group. There's the alpha keto group right here. So here is alpha ketoglutarate's R group, and that, of course, is the R group of glutamate. So again, we've traded R groups in our transamination reaction. So here is our R group for the alpha ketoglutarate, and that becomes transferred from another amino acid to another alpha keto acid, and we get glutamate. And we can, again, get glutamate from a single transamination reaction. From glutamate, it is possible for us to synthesize glutamine, proline, and arginine. So it's important to note that we can link transamination reactions in other ways. So for example, we can synthesize serine, cysteine, and glycine from 3-phosphoglycerate. So you might recognize that as a glycolytic intermediate. So the way we do that is that 3-phosphoglycerate is oxidized to 3-phosphohydroxypyruvate. So if we look over here, so now we have an alpha keto acid. There's the acid, and here's the alpha group right here. So then here's the R group. So if we do a transamination reaction, say with glutamate, then glutamate will produce the new alpha keto acid, alpha ketoglutarate. And what we get when we trade R groups now is 3-phosphoserine. So removing the phosphate gives us serine. So it's a three-step pathway, and the transamination reaction is involved in the second step. And then from serine, there are other pathways where we can synthesize glycine and cysteine. So that was sort of a whirlwind tour of really amino acid synthesis with a little bit on nitrogen fixation and assimilation. Protein synthesis, of course, occurs on ribosomes. So once we have our nice collection of amino acids, we can attach them to transfer RNAs and make a protein on the ribosomes. And we could also talk about nucleic acid synthesis, which is covered in chapter 18. But as our time is short, we will leave that for another day, and we will instead turn our attention to catabolism. So in the "Where Are We" section of our catabolic pathway map, proteins are degraded using protease enzymes that we've talked about earlier in the class, and then we, of course, get amino acids. If we remove the NH3, then we'll get some carbon skeleton, which we can use to feed into this citric acid cycle or other pathways. We should also keep in mind, even though that's beyond the scope of today's talk, that nucleotides, the purines and the pyrimidines, are broken down, and there's a way to excrete the nitrogen, but acetyl-CoA and other citric acid cycle intermediates are also produced in those pathways. So here we are back in our pathway that I've showed many times. So since we're focused on proteins, our proteins are going to be broken down into amino acids. Nucleic acids are broken down into nucleotides. So these differ from other biological molecules is that they have nitrogen. So when we degrade them, we need to be able to get rid of the nitrogen and convert it into a form in which we can excrete it. So nitrogen is essential, but too much is toxic. When we do that, we feed this into 2 and 3 carbon intermediates. Of course, that's the 2 carbon intermediate referred to here is acetyl-CoA, whereas the 3 carbon intermediate is pyruvate, but there are other intermediates in the citric acid cycle where breakdown products of amino acid and nucleotide catabolism can feed into. So before we talk about breaking down amino acids, let's talk about intracellular protein degradation. So it's interesting, cellular protein is constantly turning over as cells synthesize protein and degrade them to amino acids. So this is something that happens just as a natural course over a day. This is an interesting article. The article was a little old, but the science is still sound. But it's in an amusing way. The author writes, "Every minute of every day, a scene straight out of an Indiana Jones movie plays out in all of our cells. 1 second, a hapless protein is tooling along, just trying to do its job. The next instant, it is branded for destruction and gets sucked into a dark tunnel where it is quickly cut to pieces. Unlike Indiana Jones, there is no escape. Inside the chamber of doom, the protein is stretched out like a medieval prisoner on the rack and fed through a series of enzymatic knives that deliver the death of 1,000 cuts." Okay, let's see if we can take this amusing paragraph and translate it into the science. So first, what's the chamber of doom? Well, the chamber of doom is the proteasome. So what we want to do here is we want to distinguish it from the lysosome. So the lysosome is an organelle. It degrades proteins released from tissues that atrophy during fasting. It also is involved with disposing of obsolete parts of the cell, which is called autophagy. But again, that's beyond the scope of what we're going to talk about today. So the proteasome is not an organelle. It is, that's the chamber of doom, and it looks, so what happens here is that if we have a target protein, the target protein is branded for destruction by the attachment of a small protein called ubiquitin. So ubiquitin here is shown as little circles. This is what it really looks like over here on the right. So this tags the protein for degradation, and here is what the proteasome looks like. So here's the chamber of doom. It's a barrel, and inside the barrel are the proteolytic enzymes, and and this also has caps. So let me get rid of my pen tool right here for a second, and let's link to this video, and let's see how this works.
Protein here is ataxin 1. For ubiquitin to be carried to proteins, it needs a carrier. It's called the ubiquitin carrier enzyme. Once our ataxin has done its function and it's ready to be degraded, another enzyme will come called the ligase, the ubiquitin ligase. It comes and attached to it. And the ligase will interact with the carrier and take the ubiquitin from the carrier back to our protein. And now it can put more and more ubiquitin molecules. The protein will have to have multiple ubiquitin molecules to be degraded. And now, once it's flagged, the proteasome recognizes it, ready for degradation. The proteasome has a cap. The cap opens, the protein unwinds and enters the business end of this proteasome, the catalytic core that chops this protein into little pieces. And this protein will be degraded, and the peptides and amino acids recycled. So this is really what happens for many, many, many cellular proteins. This is a very important process. These are the players. Now imagine mutant ataxin 1. It has a different shape, and it's getting ubiquitinated. It gets to the proteasome, but its shape is so unusual. It's just not as flexible, perhaps. We think, at least, that maybe what's happening. It just can't quite unwind and get through that proteasome. It's stuck. And that what we think is happening, because at least based on some data in looking at tissues and cells, we had some evidence in support of this.
All right, so so this shows how the proteasome works. I had some students a few years ago that were quite enamored of this SpongeBob character meme, in which both of the SpongeBob characters are acting as ubiquitin, and the casket is the proteasome. It is spelled wrong, but sort of like saying to the protein, "Okay, get in." So this again describes how the proteasome works. So if I just give you some round numbers in terms of protein turnover, Americans eat about 100 grams of protein a day and probably excrete that much in nitrogen, about again up 100 grams worth a day in the urine if you're in nitrogen balance. But 400 grams per day is being turned over. So you're turning over not just the 100 grams that's in your diet, but the 300 grams that you already have endogenously. So it seems kind of wasteful. Why are proteins, why are they broken down just to synthesize them again? Because a lot of energy is required to synthesize proteins. Well, maybe it could be looked at as wasteful, but it's an important biological process. So here are a couple of reasons why you'd want to have protein turnover be an active part of metabolism. So one is that you can regulate enzyme activity. So we talked about how, in this class, how you could phosphorylate or use allosteric activators or inhibitors to control enzyme activity. But you can control enzyme activity just by degrading the enzyme or degrading an inhibitor. That's really important in the cell cycle. There are certain situations, starvation, advanced cancer, AIDS, in which protein degradation produces amino acids which can be used for energy. And this is from that same article, which I find interesting. Our immune system, in its search to eliminate virus-infected or cancerous cells, depends on the proteasome. In this process, the immune system functions like a suspicious landlady, checking whether her tenants are doing something undesirable by monitoring what they throw out in their daily trash. So somehow the immune system is able to look at foreign invaders and it is able to help fight them off using the idea of this protein degradation. So this is an overview of amino acid catabolism, and I want to use this to focus on the rest of today's talk. So the amino acid has to be broken down into its carbon skeleton, and the amino group becomes an ammonia. The ammonia is converted to urea. That carbon skeleton, where does that go? Well, it depends on the nutritional state and the needs of the organism. You can use that carbon skeleton for energy. You can oxidize it to CO2 and water and ATP. If the organism needs glucose, then if you are in the liver or the kidney, then you can carry out gluconeogenesis. The carbon skeleton can be broken down into acetyl-CoA that could feed into the citric acid cycle. If you have OAA, and if you don't, then the excess can be used to make ketone bodies. So there are lots of different choices for the carbon skeleton depending on what that is, and that depends on what the amino acid you started with. So this shows different ways that organisms excrete nitrogen. So ammoniotelic organisms are aquatic. They can excrete ammonia directly into the water because NH3 is water soluble. We are ureotelic organisms in which the vast majority of the nitrogen that we excrete is converted to urea. So we should know that that happens in the liver. And then travels to the blood to be excreted in the urine. Birds convert NH3 to uric acid. That's a solid. So that's the lovely white solid you find on different places. So that's uric acid that's excreted by birds. But that helps them conserve water because they can excrete it as a solid. So there are 3 steps that we want to talk about in how we're going to accomplish amino acid breakdown. So first is a transamination reaction. So here I have an amino acid, and I have a blue box around the R group, and then I have alpha ketoglutarate, and its R group is boxed with yellow. And if we carry out a transamination reaction, we will trade, and we will get a new alpha keto acid, which one depends on the amino acid we started with, and we will get a new amino acid, and that's glutamate. Step 2 is also a transamination reaction. We can take glutamate and we can take oxaloacetate as our alpha keto acid, and then do a second transamination reaction to get alpha ketoglutarate and aspartate. The third step is oxidative deamination. So in oxidative deamination, what happens here is this hydrogen and this ammonia, if you see that they're different colors. So the hydrogens, the protons and the electrons are donated to either NAD plus or NADP plus to form NADH or NADPH. And we lose that NH3 group as an ammonium ion, and this is a hydrolysis. So this is a hydrolytic deamination. So this shows the intermediate right here. So we can convert glutamate into alpha ketoglutarate plus the ammonium ions. So the 3 steps that I just showed you, I've outlined here. So the first step was the transamination reaction involving just any amino acid. The second one was also a transamination reaction involving the glutamate you produce in the first step, and OAA, to form alpha ketoglutarate and aspartate. And then the second reaction involved the hydrolytic deamination of glutamate to form alpha ketoglutarate. So what I did here is I took all of those 3 steps and I added everything up, and I crossed out everything that's on both sides. And this is what I get. So on the right hand side, we have an amino acid, glutamate, OAA, water, and NAD plus or NADP plus. So that's the amino acid that we are deaminating to ammonia and an alpha keto acid. Over here, that alpha keto acid that's the product can serve as a precursor to other compounds. And the NH4 plus, the ammonium ion, has to be converted to urea for excretion. So all of these other players right here can just be returned to the cellular pool. But that's the essence of what's happening. Our amino acid was deaminated to form NH4 plus and an alpha keto acid. So those 3 steps are diagrammed up here on top in this diagram. So the NH3 or NH4 plus, depending on the protonation state, the next step involves the condensation of that with bicarbonate in the carbamyl phosphate synthetase reaction to form carbamyl phosphate, and then carbamyl phosphate enters the citric acid cycle. And in so doing, we will convert these NH3 groups to urea. Again, note that the urea cycle occurs in the liver. The urea, once it is formed, leaves the liver and travels via the bloodstream to the kidney, and then it's excreted in the urine. So let's look at this carbamyl phosphate synthetase reaction. Okay, so this is how we get into the urea cycle. So we have ammonia plus bicarbonate plus 2 ATPs, and the enzyme is carbamylyl phosphate synthetase 1. There is a carbamylyl phosphate synthetase 2, but that's involved in pyrimidine biosynthesis, which we're not going to talk about today. And then we end up with carbamylyl phosphate. So over here, this NH2 group right here, that came from the ammonia, that came from the deaminated amino acid. And this group right here came from the bicarbonate. And then we have a phosphate group that came from one of the ATPs. So an overview of what happens is once we make this carbamylyl phosphate, we will carry out a reaction in which the carbamylyl phosphate will condense with ornithine to form a compound called citrulline. And in another step, we will involve an aspartate, which will donate a second nitrogen. So that aspartate could have been the product of another transamination reaction of another amino acid. So what we'll want to do when we look at the urea cycle reactions is we'll want to keep track of our nitrogens. So let's go through the steps in more detail. Here's our carbamylyl phosphate, and I colored the NH2 group in blue. So remember, that's the amino group that came from NH3, that came from the amino group of a deaminated amino acid. So the carbamylyl phosphate plus ornithine condensed to form citrulline. So again, I kept this colored blue because I want to keep track of my nitrogens. So this is the enzyme is ornithine transcarbamylase. The second reaction involves our citrulline and aspartate. So here I colored the aspartate NH3. I colored that in blue too, just to remind ourselves that that would have come from another amino group of another amino acid from a transamination reaction. And we form this huge molecule here called argininosuccinate. So that's an uphill reaction. So we'll need the ATP, which we'll need the hydrolysis of 2 phosphoanhydride bonds here to drive that reaction. So here the enzyme is argininosuccinate synthetase. So the next thing that happens is we will carry out the breakage of this bond right here. And when we do that, we should we form 2 molecules that hopefully should look familiar: arginine and fumarate. So fumarate can be converted to OAA and enter gluconeogenesis, and this is arginine, which is the precursor of urea. So you can see right here that and I colored the carbon in blue too. So that's what's going to be our urea. So in the very last step, we're going to do a hydrolysis of this bond right here, and we release urea, which I've shown in blue, and these carbons right here, that is ornithine. So we have regenerated our ornithine, and we can begin the cycle again with another molecule of carbamylyl phosphate that came from ammonia, CO2, and ATP. So I'm going to take us to this animated figure right here. And I like this figure because, let's see, I need my flash player to do this. All right, I have to, my flash player is blocked, so let's us go ahead and tell it that it is okay, that we want the flash player to work. All right, so now this should work if I hit allow. Okay. So it's the screen is going to be cropped a little bit, so I apologize for that. I had set it for the PowerPoint, and it's a little bit short. So, but here we should make note of the fact that the carbamylyl phosphate synthetase reaction is taking place inside the mitochondrion. Okay, so here we have our carbamylyl phosphate, and ornithine will enter the mitochondrion via a specific transporter, and then we can accomplish that first reaction where ornithine and carbamylyl phosphate condense to form citrulline. And what happens next is that citrulline is going to leave via a specific transport and go into the cytosol. So citrulline is now in the cytosol, and now we will use a molecule of aspartate, and we will carry out this next synthesis right here where we get the argininosuccinate. And here we can see the beginnings of our urea. right here. And then next we're going to break this bond right here, and then that leaves us with fumarate. And I'm sorry that this is cut off on your screen, but the next, the compound that's over on the left side of the screen is arginine. What they're showing over here is that fumarate can be converted to malate, and then malate can be converted to OAA, and then OAA can be converted to aspartate into, by another transamination reaction. And over here we can see with these transamination reactions, this is how the other amino acids feed into this cycle. Okay, so meanwhile, we have fumarate and we have arginine. Arginine will continue along the urea cycle pathway. We will break this bond right here. We'll do a hydrolysis and we'll release urea, and that regenerates ornithine, and now ornithine is transported into the mitochondrial matrix to continue the cycle. All right, so that is the end of the cycle. So we're back to our PowerPoint now. So those reactions are all diagrammed here in the PowerPoint that I posted. This link to this animated figure is live, and it's also posted on Sakai if you want to go back and look at it again. So we want to talk a little bit about how the urea cycle is regulated. So one of the ways we can regulate is this carbamyl phosphate synthetase 1, because that is the enzyme that synthesizes bicarbonate and the ammonia. Again, the ammonia came from deaminated amino acids to form the carbamyl phosphate, and that's what starts the urea cycle. So it turns out that this carbamyl phosphate synthetase 1 is allosterically activated by N-acetylglutamate. So we can make N-acetylglutamate from extra glutamate. So if you look at this reaction here in the box, here's glutamate and here's acetyl-CoA. So acetyl-CoA is the acetyl group donor. So there's our N-acetylglutamate. So when N-acetylglutamate concentrations are high, that means you have extra glutamate, and that means that if you have extra glutamate, if glutamate concentrations are high, then amino acid deamination rates must be high. And that's going to produce a lot of NH3, and you want to be able to incorporate that NH3 into urea for excretion, because too much NH3 would be too basic for the blood, and that would not be safe. So again, if I go back to my three-step reaction right here, again, I did my 3 steps, and I crossed out everything that was the same on both sides. So we took our amino acid and we deaminated it, and we'll be left with a carbon skeleton, which is a precursor to other compounds. And we just talked about how that ammonium ion is converted to urea for excretion. So we've already talked about this. So let's go back here and talk about this alpha keto acid. So what happens to that? Well, we're going to convert it to other metabolites, and depending on what the amino acid we start with will depend on what alpha keto acid we get, and we can describe the amino acid then as glucogenic or ketogenic. So a glucogenic amino acid, when the amino acid is deaminated, form the NH3, forms this carbon skeleton. And if the carbon skeleton can get to pyruvate or some other citric acid cycle intermediate, as long as you have 3 carbons, a minimum of 3 carbons, then you can get to gluconeogenesis, and you can get to glucose that way. So this would happen if you were on a diet that was very high in protein and very low in carbohydrate, because our brain needs glucose and our body finds a way to get glucose any way it can, even if you're not eating it. So a very high protein diet would produce this kind of reaction. Also starvation, if you were sacrificing some of your body protein to get glucose that way, if glucose wasn't part of the diet. We also have amino acids that we refer to as ketogenic. So these produce carbon skeletons. Either we go to acetyl-CoA, which is converted to ketone bodies, or maybe we don't even go to acetyl-CoA, the carbon skeleton just goes right to the ketone body. So remember, acetyl-CoA has 2 carbons, and there isn't any way, at least not in mammals, where you can get you can't get to glucose from acetyl-CoA. You don't have enough carbons. 2 carbons won't work. So if you have the acetyl-CoA, that can go into the citric acid cycle if you have enough OAA. But if you don't have enough OAA, because OAA is being diverted to gluconeogenesis, then the extra acetyl-CoA is converted to ketone bodies, and that can be used for energy by some body tissues. So if we look at all of the amino acids that we have here, I just put green boxes around. These amino acids are glucogenic. So if I can be degraded from pyruvate or OAA or fumarate or succinyl-CoA or alpha-ketoglutarate, these are citric acid cycle intermediates. I can somehow get my way to OAA, and then I can use the pyruvate carboxylase reaction to enter the gluconeogenic pathway. Again, that happens in the liver and kidney. But the amino acids with the pink boxes, either they are converted to ketone bodies directly, or they're converted to acetyl-CoA, which is converted to acetoacetate, which is a ketone body, if oxaloacetate is in short supply. So if you look at all of these amino acids, we can see that only leucine and lysine are exclusively ketogenic. Really, all the others are both, because if you can get to pyruvate, then you can get to acetyl-CoA. It's just the reverse is something that you can't do, at least not in mammals. So if we are going to go through a whirlwind tour of amino acid catabolism, this shows how we can take these pathways involving glycine, serine, alanine, and cysteine can all be broken down into pyruvate. So all of these amino acids are glucogenic, because if you can get to pyruvate, you can get to OAA, and if you can get to OAA, you can get to glucose. Here's another example of amino acid catabolism. So we can deaminate asparagine, and the NH4 plus would have to go through the urea cycle to aspartate, and then we can go through a transamination reaction, and we can get to OAA. If you get to OAA, we can get to glucose. Arg, glyn, his, and pro are all converted to glutamate. So just as an example, glutaminase catalyzes the hydrolysis of glyn to glue in ammonia. So, and if all of these amino acids can find their way to glutamate, then glutamate, as we saw earlier, can be converted to alpha-ketoglutarate, either the glutamate dehydrogenase reaction or a transamination reaction, and then that can enter the citric acid cycle. And again, if you can get to alpha-ketoglutarate, you can get to OAA, and you can get to glucose. So these are all examples of amino acids whose breakdown products can feed into gluconeogenesis. So more examples. Ili, met, and val are the branch chain amino acids, and they're degraded to propionyl-CoA, and then that can be converted to succinyl-CoA in a reaction that requires vitamin B12, and then succinyl-CoA can enter the citric acid cycle. And in a way that sort of brings us full circle from where we started at the beginning of the semester with Chapter 2, in the kidney, there are a series of pathways where glutamine is used to synthesize bicarbonate. So we get glucose here, of course, because the kidney is a gluconeogenic tissue. And here, we can see how the kidney is involved with the regulation of blood pH. And we saw earlier in Chapter 2 how we could use exhalation of CO2 in the lungs, and that influences the equilibrium between carbonic acid and bicarbonate. But the kidney plays a very large role in regulating pH. So by regulating either the reabsorption of the excretion of bicarbonate ions and ammonium ions, the kidney can play a very large part in regulating blood pH. So I just wanted to leave you with one more example of amino acid catabolism, because I thought you would find this interesting. So phenylalanine, there's an enzyme called phenylalanine hydroxylase, and it converts phenylalanine to tyrosine. And then there are a number of steps in which we get acetoacetate, which is a ketone body, and fumarate, which can be converted to OAA and then to glucose. So what that means is that phenylalanine is both ketogenic and glucogenic. It's ketogenic because it forms a ketone body, but it's glucogenic because it's converted to fumarate, which gives you 4 carbons, which is enough carbons to be converted to glucose. So this is the reaction. So phenylalanine is converted to tyrosine. So the enzyme is phenylalanine hydroxylase, and that describes exactly what it does. And hopefully you remember that tyrosine looks like phenylalanine except with the hydroxyl group. So I thought you would find this interesting is because I know a lot of you are interested in genetics, and phenylalanine hydroxylase is missing in patients with PKU, which is phenylketonuria. So then the disease gets its name because phenylalanine accumulates in the blood and it undergoes a transamination reaction and it forms a phenylketone. So again, this is an alpha keto acid right here. There's the acid, there's the ketone, and then there's the R group of phenylalanine. And when I was a biochemistry student many moons ago, I was taught that the phenylketone causes brain damage. Um, so and that brain damage is irreversible. So that's why babies are screened for PKU at birth. So there's just a little heel prick, which is shown here up on the top, um, and so babies are screened for PKU at birth. So at the bottom, there's a young man who has been diagnosed with PKU, and he is making himself a protein shake. He needs to be able to monitor his phenylalanine intake very, very closely because he needs just enough phenylalanine to provide the amino acid that's needed for protein synthesis, but he can't have any extra. So some of the articles I've been reading recently suggest that it might be the phenylalanine itself that can cause damage to the brain. This was a paper that came out a while ago, and I haven't heard what the follow-up is to this, but I thought this was interesting. This is from Chemical Engineering News that showed that phenylalanine by itself can form aggregates in the brain that look like amyloid fibrils. And this is interesting because this isn't a protein. This is just a collection of phenylalanine molecules, but somehow they associate to form these amyloid-like fibrils. So this is something that is similar to what you'd see in Alzheimer's disease, for example. So maybe it's not the phenylketone at all. Maybe it's the phenylalanine itself that has this kind of impact on the brain. So somebody with PKU cannot have any extra phenylalanine because these are the kinds of effects that you could have. So that is our whirlwind tour of amino acid catabolism. I hope you enjoyed it, and I will see you next time.